So, good morning, good afternoon, everyone. My name is Crystal Floridinti, and my partner is Kristamita Mulaki. We'll be seeing her in the next few slides. So, our research or our topic is assessment of the potential for cross contamination of food products by reusable Ziplocs. So, we came up with this topic because we have a friend who's always using a Ziploc for her food. So, we actually kind of interview her with how she used the Ziploc. So she told us that sometimes it still depends on the content. Sometimes she uses it without washing, and sometimes when it comes to very wet food like marinated food, um, food that is very moist, she would reuse it still. However, she would wash it. So actually, in our household, my partner and I, we use Ziplocs a lot. We reuse them as well, especially with the food that we want to marinate. The thing is, this Ziplocs are very convenient to use. It has different sizes and everything. You can store different food products, and you can store it in the fridge. You can store it in the freezer. Everywhere you can use Ziplocs. So got really curious about the possible contamination or possible cross contamination the Ziplocs were using. So for the objectives of the study, we want to figure out or we want to determine if there is a microbes in this reusable Ziploc. So if microbes are presently growing there. Second, of we want to know the count or the concentration of these microbes that were present on the reusable Ziplocs. Third, we want to know which microorganism are growing in this reusable Ziplocs, and we also want to know which organism is predominantly growing. So lastly, we also want to know if washing is enough or is washing efficient in this reusable Ziploc. So if it's okay to reuse the Ziplocs after washing them. So basically, after knowing the objectives of this study, we come up with the importance of the study. So readers will be aware of the possible contaminants of reusing the Ziplocs, and this will lead to the safety of the public or the users of Ziplocs. So here are some of the related topics or related articles rather um, into our research, and the link that is posted above this three images is one of the main research that we found on the internet in relation to our topic. So we were trying to look for a specific topic or a specific research that is conducted in Ziplocs. However, we only found food containers, which are plastic containers, Tupperwares, takeout boxes, containers, and also some of the studies that helped us to formulate or to create the methods, the objectives, the hypothesis is the containers conducted or the research conducted on some of our water bottles. So we correlated our topic with that too. So just to brief you with the articles that help us with our paper. So there are still debates whether reusable Ziplocs is safe, and we found out that it still depends on the content and how the users is effectively washing them and how they store them. So in a few slides, you'd get to know our results and how our conclusions or how our final decision is made. So what I would suggest is you try looking into these articles, and if you're interested, you can read them. So for the methods, we actually divided it into four parts. The collection of the Ziplocs, bacterial assay enumeration, bacterial identification, and we want to know the effectivity of washing as well. So for the collection of Ziplocs, we actually considered the state of the Ziploc, if is it thoroughly used, if it's brand new, how many days it was used, and was it washed before we sampled it, the contents of the Ziplocs as well, and how the user stored it. For the bacterial assay enumeration, we did dilution from the Ziplocs, from the inside of the Ziploc, and then we transfer this dilution to spread plate. For the identification of bacteria, we did we did gram staining first to identify whether it is a gram positive or a gram negative, and we plated it into their specific plates. We used three of the plates, hectowin, mannitol salt agar, and McConkey. And we also identify if washing is effective, or would it be recommended to reuse a Ziploc after washing. So for the collection of Ziplocs, we actually in total we collected 17 Ziplocs, and we tested out using dilution. We used the normal saline solution. It's actually an isotonic solution for recovery of bacteria. So we use for the spread plate, we use the trypticase soy agar. It's a nutrient agar which help with the growth of the possible contaminations that we are expecting. And after dilution and after spreading them on the spread plate, we incubated these plates um, after 18 to 24 hours. So for the bacterial identification, what we did is from the TSA spread plate, we identify different colonies, and then we have them gram stained to identify whether if it is a gram positive or if it is a gram negative. So we can plate them on their respective medias. So we actually use three medias here, hectowin, McConkey, and mannitol salt agar. For the gram positive, we plate it to MSA or mannitol salt agar. For the gram negative, we plate it into hectowin and McConkey. The reason is we thought of the relevancy of what organism will grow into these medias. We have considered Salmonella and Shigella, E. coli, and Staphylococcus aureus, which is relevant to food products or food containers because it causes food illness. So after identifying the bacterial growth on the TSA, we proceeded with the washing. We also wanted to know whether if it is effective or it is there are different ways of washing or cleaning Ziplocs. So we came up with three ways of washing Ziplocs. So first one is we wash the Ziplocs using a washing or a dishwashing liquid that can be found on the grocery. We did this for all the washing of Ziplocs. First method is we wash it and then we just leave it for 1 week. Store it without drying. Second one is we wash it and then we dry it after 30 minutes of washing with paper towels. And then the last and third method that we thought of was washing the Ziplocs and then having it dried immediately after washing with paper towels. So from these three methods, after doing this, we did dilution as well, and we plated them to spread plate to identify the concentration or the number of bacteria that will grow.
So for the result of our swab on October 16, 2023, These items, which is the marinated raw meat stored in fridge, raw meat stored in freezer, vegetable, which is the cabbage, and the cooked food. So these items were chosen to simulate various potential contaminating factors that could affect the Ziploc bags during the regular use. So the intention behind using these materials was to mimic realistic scenarios and evaluate the resilience of the Ziplocs against the potential contact with food items in different stages, which is the raw, marinated, and cook, along with the fresh vegetable. So the methodology employed aimed at assessing the durability and cleanliness of the reusable Ziplocs in everyday scenarios to ensure their suitability for safe food storage. So uh, for the marinated for the marinated raw meat, we obtained the result from 10 raised to negative 4, which is T and TC, or two numerals to count, and from 10 raised to negative 5, T and TC as well, and for the 10 raised to 6, we got T and TC as well, and we ended up with a result of 2.0 times 10 raised to 9. So for the marinated raw meat, the Ziploc bags used for storing marinated raw meat exhibit um, exceptionally high microbial counts. It indicates potential contamination. So the possibility of cross contamination between the bags and the stored food is significant. And for the raw meat, which is stored in the freezer, we obtain the result of from 10 raised to negative 4, we got 10 colonies, and from 10 raised to negative 5, we got 4 colonies, and from 10 raised to negative 6, we got 3 colonies, and we got the result of 1.0 times 10 to raised to 6. So for the raw meat, which is stored from the freezer, it is moderate microbial counts were observed on the Ziploc bags storing the raw meat. So while lowering the marinated raw meat, there is still a potential risk of cross contamination if not addressed. And for the vegetable, which is the cabbage, we obtain the result from the 10 raised to negative 4, which is TNTC, and for the 10 raised to negative 5, we got 150 colonies, and for the 10 raised to negative 6, we got 60 colonies. So we ended up with a result of 7.0 times 10 raised to 9. So the presence of high microbial counts on Ziploc bags used for vegetables suggests a high potential for cross-contamination. And for the cooked food, um, we obtain the result of from the plates that so for the cooked food, from the plate, which is the 10 raised to negative 4, we got 1 colonies, and from the 10 raised to negative 5, there is no growth, and for the 10 raised to negative 6, still no growth. So possible contaminant minimal counts were observed on bags used for storing cooked food. It indicating a lower risk, but the presence of possible contaminant highlights the need for caution. So, for the result of our swab on October 16, 2023, the result strongly suggests that the reusable Ziploc bags might be contributing to cross-contamination, especially when used for storing raw and marinated meats and vegetables. Proper cleaning and sanitation, and potentially considering the replacement of a heavily contaminated bags are recommended to avoid to avoid the cross contamination. And for the result of our swab on October 30, 2023, during the testing process, the following items were used: the tofu, butter, cooked chicken, and a previous use Ziploc bag that was not washed and had been stored for a couple of months. Okay. These items were deliberately selected to represent various potential contaminants that the Ziploc bags might encounter during their typical usage cycle. The intention behind using tofu, butter, and cooked chicken was to simulate different food types and textures that might come into contact with the Ziploc bags. And additionally, the inclusion of the previous use and unclean Ziploc aim to assist the aim to assess the effects of a prolonged storage on potential contaminants. So, for the result, for the tofu, from 10 raised to negative 4, we got one colony, and for the 10 raised to negative 5, there is no growth, and for the 10 raised to negative 6, still there is no growth. So, for the result, we ended up with 1.0 times 10 raised to 5. So for the tofu, minimal counts were observed, indicating generally acceptable storage condition, but a slightly higher microbial presence compared to some other items. And for the butter, from 10 raised to negative 4, the result is no growth. And for the 10 raised to negative 5, still no growth. And for the 10 raised to negative 6, still no growth. And we got the result of less than 1.0 times 10 raised to 4. So for the butter stored in the Ziploc bag exhibited negligible microbial count, reflecting a satisfactory storage condition. And for the cooked chicken, we got the result from 10 raised to negative 4, we got 23 colony, and from the plate, which is the 10 raised to negative 10 raised to negative 5, we got 2 colonies, and for the 10 raised to negative 6, we got no growth, and we got the result of 2.3 times 10 to 10 raised to 6. And for the cooked chicken, uh, we observe a moderate to high microbial count were observed, and indicating a potential risk of a contamination, particularly considering the presence of cooked chicken. And for the Ziploc use without washing, from 10 raised to negative 4, it is TNTC, and from the 10 raised to negative 5, it is TNTC, and for the 10 raised to negative 6, we got 158 colonies, and we got the result of 1.6 times 10 raised to 9. So for the Ziploc bag itself, exhibited extremely high microbial counts, suggesting a significant potential for cross-contamination due to extended storage without washing. So, um, overall, the results indicate varying levels of microbial presence in different items storage within Ziploc bags. Items like raw meat and the Ziploc itself showed particularly high counts, indicating a notable risk of contamination. So, In conclusion, proper storage, especially for items prone to microbial growth, is crucial. So, immediate actions such as discarding highly contaminated items and replacing the Ziploc bags, or thoroughly washing and sanitizing them before use, are recommended to mitigate the contamination risks. So for the result of our swab on November 6, 2023, we got the result of invalid. It is because of all of our samples are contaminated due to improper storing of the plates after incubation. So here is a result of our Ziploc that is being washed, and no drying means no paper towel being used, and it is stored for 1 week. So as you can see, for the first Ziploc, which is the marinated raw meat, it shows TNTC from the plate, which is the 10 raised to negative 4 and 10 to negative 10 raised to negative 5, while on the 10 raised to negative 6 it shows 136 colonies, and upon doing the gram stain it shows gram positive bacteria. And for the media, we use the mannitol salt agar for our biochemical testing. So the explanation for the result of marinated raw meat, despite washing, the previously contaminated Ziploc bag retained a substantial microbial presence on both gram stain and MSA media, it is suggesting a potential inefficiency in the washing method. And for the raw meat, which is stored in freezer, it has one colony on the plate, which is the 10 raised to negative 4, while on the 10 raised to negative 5 and to 10 raised to negative 6, it shows no growth. So the gram stain and the biochemical testing were no longer performed. And as you can see from the vegetable, which is the cabbage, from the plate 10 raised to negative 4 up to 10 raised to negative 6, it shows no growth as well. So the gram stain and biochemical testing were not performed. So the explanation for both the raw meat and the vegetable Ziploc bags, which initially showed no growth on TSA, continued to exhibit no microbial growth even after washing and subsequent testing on gram stain and MSA media. So our conclusion for this is procedure. The experiment indicates that while washing might eliminate contaminants from Ziploc bags, it might be less effective for heavily contaminated ones. So, as observed in the marinated raw meat bag, further refinement of washing method or alternative sanitation measures may be necessary for heavily contaminated bags. So for this procedure, the Ziploc wash and dried after 30 minutes of standing. And so here's our result. For the raw meat, it shows no growth from the plates 10 raised to negative 4 up to 10 raised to negative 6. And we didn't do a gram stain and biochemical test since it shows no growth. And for the cooked chicken, it has two numbers to count, or TNTC result from 10 raised to negative 4 up to 10 raised to negative 6. And we did the gram stain, and it shows positive. It shows gram positive bacteria. And for the biochemical test, it shows positive as well. And we use a MSA media. And for the Ziploc use without washing and stored for a couple of months, it shows no growth from 10 raised to negative 4 up to 10 raised to negative 6. Okay, so overall, after washing the raw meat bag, exhibit no microbial growth on both gram stain and MSA media, indicating effective removal of contaminants. However, the previously heavily contaminated cooked chicken bag still showed a substantial growth on gram stain and positive growth on MSA. indicating persistence of contaminants or inadequate washing. So here's the result of our Ziploc that washed and dried immediately with paper towels. So as you can see, all of our plates from 10 raised to negative 4 up to 10 raised to negative 6. It shows no growth from all the plates. So gram stain and biochemical testing on medias were not performed anymore. So this result indicates that the immediate washing of previously contaminated Ziploc bags effectively eliminated microbial growth, as evidenced by absence of a growth on gram stain medias across all the tested samples. This outcome indicates that the washing method applied were successful in removing contaminants from the bags promptly. And immediate washing and drying with paper towels proved to be successful approach in preventing further microbial growth on the previously contaminated bags. So here's the result of our plates that has growth on medias. So here are the conclusions regarding the reuse of Ziploc bags and potential contamination. First is the economic benefits of reusing Ziploc bags. So reusing Ziploc bags is economical as it reduces the need for frequent replacements, saving money and reducing waste. This conclusion stems from the understanding that a simple bag can reuse multiple times for various purposes. And the second is the potential contamination of reused Ziploc bags. Upon testing, there might be a contamination found in the reused Ziploc bags. This suggests that despite attempts to clean or wash them, contaminants might still present. Contamination could stem from various sources such as food residues, environmental exposure, and or improper washing techniques. And third is the incomplete elimination of contaminants by washing. So, washing alone might not eliminate 100% of present contaminants from the Ziploc bags. Um, this conclusion is based on the understanding that some contaminants might adhere to the surface or remain within inside the bags even after washing with soap and water. And the fourth is the factors affecting the effectiveness of washing. Several factors can influence how effectively contaminants are removed from the Ziploc bags during washing. Factors like water temperature, type of detergent, or cleaning agent used, duration of washing, and the material of Ziploc bags could impact the efficacy of the cleaning process. And the last one is the fifth one is the importance of public awareness, especially for the immunocompromised person. It's crucial to raise awareness among the public immune system about the potential risks associated with reusing Ziploc bags. These individuals are more susceptible to infection, and using potentially contaminated bags could pose health risks. And the last one is need for further study. Given the complexity of the issue and the potential health implications, further research is warranted. This could involve detailed studies on the effectiveness of various cleaning methods, the type of contaminants commonly found on reused Ziploc bags, and the impact of contaminants on different populations, especially those the weakened immune systems. Each of the conclusion highlights the importance of understanding the risks and limitation associated with reusing Ziploc bags and emphasize the need for proper hygiene practices and further investigation to ensure public health and safety. Thank you.